Okay, why don't we go ahead and get started? Thank you all for coming. There's a number of you I don't know. My name's Bill Hirsch. I'm chair of the department and overall director of the program, although Dr. McQueenie directs the bioinformatics and computational biology track. So we're here today for the dissertation defense, always an exciting time for a student who's completing their studies, hopefully completing their studies. I have good confidence in Eisa. So Eisa Mahyari has been a student in our program. I assume most of you in the room know him. He originally obtained bachelor's degrees in biology and chemistry from Portland State, obtained a master's degree in biochemistry and molecular biology here at OHSU, and then he joined our program. Prior to joining our department, he worked on a large project elucidating the vaccine-induced correlated protection against a highly pathogenic HIV homolog in the rhesus macaque model. I think I pronounced all that correctly. For his dissertation, he's focused on developing a computational framework for robust identification of rare cellular subsets and single-cell technologies. So he's defending his dissertation. Just for those of you who are not familiar with our department, the way these work is that first the candidate presents his talk, usually 30 to 45 minutes, and then he takes questions from everyone. At that point, the public, the audience, except for his committee, is excused, and then there's additional questioning from his committee. Then he's excused, and then the committee makes the determination. So I won't take up any more of Eisa's time. And
thank you, Dr. Thank you, everyone, for coming. It's such an exciting time to be presenting my work to you and my research. And although this is going to be a part of the results and the work that I did, it's focused on one form of technology. So I guess we get started. So in general, we were interested in finding computational models to be able to identify rare cellular subsets from a heterogeneous mix of cells. And that's because rare cellular subsets are important clinically and for research determinants of health and disease. For example, one leukemic cell in 10,000 healthy cells is prognostic of minimal residual disease in adult lymphoblastic leukemia. And on the other side, of interest, more recent research interest, is these circulating tumor cells, where publications are showing that there are around 1 in 10 million healthy cells. So they're very important in terms of determinants of health and disease. What makes them so difficult to identify is two factors. I'm showing this in a bimodal situation. One is effect size, which is a standardized unit of difference between two populations. And the smaller that difference, the more difficult it is to define a discriminatory boundary between them and to be able to classify them as one or the other. On the other side, marginal frequency defines how many of those are present relative to the other population. And the less there are of those positive class, it's harder to make a judgment call on where to draw a boundary line, as well as it's not enough events for training machine learning or computational methods, usually. So that's where the difficulty comes in. Furthermore, in real-world applications, it's not usually a binary classification that we're interested in. Each population may be associated with false and negative positive rates. Sorry, I said that wrong. False positive or in false negative rates. There's also this idea of high dimensionality, where a lot of times we have to do this feature selection to be able to do the classification that we need. Phenotypes are not necessarily also distinct. They could be gradients, and that also makes them difficult. And more importantly, there are confounding factors in experiments, in everything that is done, that affect the classification, especially rare cellular subsets. As I said, confounders are important, impede computational methods, but it's at multiple scopes. So we have to think about standardizing protocols and experimental and analytical methods so that we can improve the classification goal that we have in the scope of this research. For example, multiplexing is one way to be able to do that, but another way to overcome such limitations that confounding factors produce is normalization. And here's a very commonly used method that I'm trying to just introduce the idea of normalization with. There are two data sets, single-cell RNA-seq data sets here, labeled A and B. And you can see on the log scale gene counts, the two are different. And these are 10 genes that I've just chosen to show that although these two data sets are similar in terms of what they're trying to represent, they are shifted, and their quantitative measurements differ from each other. And what quantitative normalization is trying to do is create, make the distributions of the two data sets similar so that we can run models that have assumptions about them having identical distributions and et cetera like that. So, and what normalization we choose actually depends on, a lot of times, the experimental design and as well as the protocol or the instrument that we're using. The instrument that we're using that I've worked on, focused mostly on in my dissertation, is single-cell RNA-seq. There are different instruments that do this. They all start with capturing of single cells. For each cell, the mRNA is quantified, captured, converted to cDNA, amplified, and then sequenced with a specific platform. Once the raw reads have been acquired, we need to do a lot of bioinformatics and these specifically defined pipelines to make sure it's the quality control it, remove any aberrant cells or unwanted debris. And in the end, we have these single specific normalizations that are done, depending on the platform again, to normalize for RNA content, library size, and the variation that's between the cells, usually caused by unwanted biological variation. But this process could be biased against our rare cellular subsets, and a lot of times the focus of these pipelines is not rare cellular subsets. And so it may look aberrant, but they are removed because they don't, for that hypothesis that was originally used, it's not in the focus. And as I said earlier, the whole motivation of my dissertation has been to find a computational method to be able to classify rare cellular subsets. And parallel to what normal human experts do as well, where they go read literature and find examples of the phenotype or the signature that they're interested in, I've also made the same assumptions where there is always some sort of a training that we can do from data to be trained on to then be able to infer on new data sets. And the training, and that assumption focuses us on a set of machine learning methods called supervised methods, where there is this training data set that the algorithm trains on and develops a classification model, and a new test set is used to predict a new classification. But in the real world, if the marginal frequency between the test set and the training set of the population of interest differs significantly, supervised methods, many of them have issues. Also, supervised methods assume that the training data and the test data come from the same source, which means that intrinsic distributions should have this idea of identically distributed, which in most applied cases it is not. And that's why I showed the normalization potentially can resolve that, but that's a big hammer in that toolbox. So transfer learning is another domain of machine learning that we can that can also be applied here. Specifically, that what's common, one of the settings of transfer learning is called transductive transfer learning, where we do some learning and we make some inference. The major difference is that in transfer learning, we're generalizing the training and adapting it to the test set. Whereas in supervised methods, that's not the case. Furthermore, transfer learning does not have an identically distributed assumption. Also, it is also, in theory, not sensitive to the difference between marginal frequency between the two training and testing data set. So in overall, that's the better domain of machine learning to be able to research. So let's look at an example of transductive transfer learning. We have some training data, which is, you can imagine this is rows of cells and columns of features, either flow cytometry, RNA-seq, and that data set can be either split or sampled to make multiple sets. Because they're non-identical, if you train on each one independently, you get a slightly different classification hyperplane. So from each one, we train the machine learning algorithm, specifically a linear SVM, to derive at a classification per each sample. Because they're slightly different, we can get a mean of the classification hyperplanes derived from all of these samples as the mean or ensemble hyperplane. Now we have 3 test data that we get to want to classify with that mean hyperplane, but because of confounding factors that I talked about earlier, the data sets could be confounded, and so the outcome is not what we predict. Now imagine I said in a bimodal situation where there's a binary classification setting, we expect to have 2 peaks in the middle of a lower density region. So where we classify, doesn't matter how many input features we have, right? It could be 20, it could be 5, whatever. But when it gets multiplied into the hyperplane, you actually, because it's a binary classification, you expect to get 2 peaks and a center in between. But confounding factors can affect the effect size between the 2 populations or cause a shift in that to cause a problem in the classification. So 2 possible ways that we can adapt this specific generalized or mean hyperplane to these data sets is the way to go. And 2 ways to do that is either by bias shift or rotate. So bias shift is, again, this is on the y-axis, which is, again, doesn't matter how many features are inputted, but once it gets multiplied into the hyperplane, you're getting that on the y-axis, right? So the y equals that multiplication of that function. So on the y, the predicted outcome, depending on where the bias is, the outcome, the mapping can be shifted as it is here. Or if it's the wrong rotation, it can affect the effect size between the 2 populations. So the idea is to find the right rotation and bias to make as a form of adaptation to the test set. So you're using the test set that you're at hand to make these to these changes. And that is actually the contribution of Li et al. In 2011 as part of their pipeline. This is an overview of it. There are a bunch of individual algorithms in the first few. I'll get a little bit more in detail. But the whole point is that this training in the beginning is then adapted to make the final inference. So a little bit more detail into the Li et al. Pipeline. So they proposed their work on flow cytometry data, which in general there's multiple samples. Each sample has multiple cells in it, or thousands of cells in it, and many conditions, many situations. So generally each one can be represented as an individual sample, each flow cytometry sample. But in RNA-seq, we usually have one large data set that has a few thousand or a few hundred cells. So we don't have multiple samples of that. So the difference, which I'll get to later a little bit more when I'm talking about single-cell RNA-seq, is that major difference. That for Li et al., they derived directly each one of these is one sample, and they derive a linear classified hyperplane from that sample. Then, because each condition, each experiment from those flow cytometry could be very different because of issues or problems or real biology, they derive a robust mean, which is less biased towards those big outliers. So once the training is complete, the next module or the next algorithm in the pipeline, what it's trying to do is correct for a major shift compared to the test data compared to all of the training. So what it's doing is it compares the location or the position of where the test data is being mapped to a median of where the trainings were mapped. So you can do a general shift near the training. So it corrects as a first step to that adaptation process. Then, once the shift is done, the algorithm defines several potential biases as candidates, so we can see which one's the best one. And it counts how many cells fall within the margin near that boundary, near each one of those biases. But as you can imagine, depending on how far these biases are defined and how big this margin is, the line that is producing them, it could be very rigidly and produce multiple local minima. Even if you smooth it with a Gaussian smoother, you might get multiple local minimas. And when you're trying to look for that expectation where it's like that low density minima between those two peaks where you want to cut the data, you might have a second option where it's biasing that gradient descent algorithm. And gradient descent is the algorithm that actually does that, which looks for these dips to find the potential minima to call as the correct bias. And so the next module of the algorithm in the Lierau is finding the correct rotation. And that's done similarly to, you define a bunch of possible rotations, you do a count of, relative to those rotations, how many cells fall within a certain margin, and that defines this kind of a curve that also has multiple local minimas, but what you're looking for is to define it as the lowest position in that, as the optimal minimum. Again, it can be, the gradient descent can be biased by any of these. Even if it's smooth, which is in this case that I'm showing the smooth. But the Lierau algorithm is not available upon request. The data set they show it is not available. So how can I test single-cell RNA-seq data? How can I evaluate rare cellular subsets with it? So the first step was to implement a baseline version based on what they published as algorithms in their manuscript. And obviously then that becomes a little different because the way I coded it in another language with the algorithms that are kind of supporting it, that are from the language that I use, causes slight variation. So it's not an exact one-to-one. But it's intended for open source release, and so any researcher and anyone can in the future be able to expand upon it and also use it. So our first aim was to implement, adapt, and validate this previously theorized transfer learning framework to classify these cellular subsets or signatures, especially rare cellular subsets, in new single-cell RNA-seq data. Once I had this built, I had some pilot data. I had some data from the Lewinson lab that I tested with, and I came up with some list of potential extensions that I could make to make the algorithm better, especially to be able to generalize across data sets, and especially to be able to focus on rare cellular subsets. So a little overview of what it looks like. So if this is a similar, I kept the same structure of the proposed algorithm. So between the baseline and the extension, the first three are the same. So we did the trainings the same, the general shift is the same, but in the extended version, how we define where the bias is and where the proper rotation is changed, as well as a new change in how the smoothing is done is also changed, because the way they proposed the smoothing, it didn't produce a lot of local minimas, and I wanted more of a smooth curve. So I even used a different way to estimate the better bandwidth to smooth the density curve. I'll go a little bit more in detail into each one of these, but on top of all of this similar structure, I had a new additional module that actually is for specifically for rare cellular subsets, and I'll get a little bit more into what that does as well in a second. So the first extension for the algorithm part 4, which is trying to find that bias, as I said, the whole idea is to be able to find a smooth line. So instead of finding multiple biases and a margin around them to do the count, I just did a direct density. But that needs to be smooth with that new bandwidth method that gives additional smoothness to the curve. From that, to derive multiple other additional evidences of topology, the first derivative and the inverse of the second derivative are computed of this density curve, which also have relationships to how the rates of increase, decrease, and all this. You can imagine, if you remember calculus, what first and second derivatives do is you're affecting the rate of change in this curve. That would be the first derivative. And the second derivative is the derivative of the first derivative, and then the inverse is over the what do you call it? In the denominator. You put it's 1 over the f of x. There you go. So in the count, I'm getting tired of talking. And I should just say that doing this directly without defining any of those biases and the margins around them removes that whole margin parameter that's in the baseline as a bias, because what you have to do is find initially the correct margin, and if you go by a default setting, you may not have the correct margin. That's actually a benefit of that. And the last part, on the finding the rotation, is a similar thing. We want to try to make it more smooth, but the counts are now different. This is no longer it's counting. It's relative to the amount of false negatives that are in the function, and you're trying to minimize the number of negative calls that it's making. And that innately is a smoother function. And then with that update on the bandwidth selection also makes it a little even more smooth to be able to find the optimal minima as opposed to a local minima. So that new module that I talked about focuses on the rare cellular subsets, works for both baseline and the extended versions of the algorithm of the whole pipeline. But basically the whole idea is when you're trying to make an inference on a rare subset, the prediction is difficult to make because there's a potentially a large number of local minimas that you have to choose from. And the whole idea is to, from the training, to get an estimate of potentially what that population's frequency is, plus some error, because we truly don't know the margin of frequency of in the test set. And that can be used to narrow the search space in the mapping, so that now we are looking for a smaller region to find that local minima, optimal minima. So once I had both the baseline and the extended versions built and tested, the idea was we need to evaluate and benchmark its performance to some other previously work done. In the next part of the aim, we want to quantify classification generalizability, which is how well does it perform when it learns from one data set and infers on another data set, sourced differently. And finally, in part C, we were interested in evaluating how well it does on the performance on rare cellular subsets. The data sets that I used, I acquired them online from a publication that has already tested 6 other classifiers, the top performing classifiers, and so I can use very parallel to the work, and whatever my results are, I can compare it directly to them. So there are 2 data sets. One is the one shown here, and you can see that not only the marginal frequency of the 3 labels, because what we're trying to do here is classify the cell cycle labels and what is known about the both data sets. So the marginal frequency of the labels that are of interest, which is SG1 or G2M, which is different across the 2. And more importantly, they're sourced from different protocols. This one's a Fluidigm C1, and this one's from Quartz-seq. So innately, these two are highly confounded and different by the protocols and the methods used. To parallel our work with the work that was done by the published work with the other classifiers, those folks, the Scialdone et al., they found 405 cell cycle genes from a curation of a bunch of other databases and so forth, and they're testing and finding the right appropriate feature selection on these two data sets. From those 405 cells, they showed their top performance with a very good signature using 40 of those genes. But before they did that, they present their work with some sort of a with a normalization technique, which they use rank-based normalization. So again, these two are not identically distributed. As I said, it's one of the hallmarks of transfer learning, and the marginal frequencies are different in the two. So let's visualize how these data sets look just holistically a little bit in terms of with principal component analysis. So here's data set 1A, and B is this one colored in green. And using the 405 genes or the 40 genes, we can see that the signature does reduce the distance between them, the 40 gene signature, but they're still separate. And they also show a general trend that they both have, that the G1 cells, the green ones, are this way. In between of each one is the G2M cells, and then the S cells, S phase cells. But like I said, the Scialdone et al., they did rank normalization. So if I'm doing a benchmarking at first, that's the one I need to focus on as well, which is I'm showing this in the center here. So rank normalization causes the two data sets to no longer have that distance between them, no longer have that shift between them. And, but still retains that general trend of where the data is in the first and second principal components. On the other side, there's another, like I mentioned, quantile normalization is another technique commonly used, and that produces a similar but a little slightly different distribution of data. And as a non-rank-based method normalization, I use the log median absolute value normalization, which has a very squishing effect. So, as I said earlier, supervised methods can be biased by and supervised and unsupervised methods, which is completely done to be able to. So I should start again. Here I'm showing unsupervised clustering, which is a method used to be able to find subtypes within a data set. And what I'm showing on top is the actual classes known labels, where the red is G1, the blue is S, and the green is G2Ms. And so we see that the normalizations affect the unsupervised clustering that is done on these cells and produces different clusters. Although it's not immediately possible, what you can see is that the groupings change based on the normalization they used. And supervised methods, which I'll show as well, are also biased by this as well, by the normalization technique. And additionally, the 40 gene signature that they found is not an optimal signature for a linear classifier, which is what I'm going to be using as part of the framework that I developed. So I first find a potential set of gene sets that would be an alternative to that 40 gene set. So what I did was, for each class label from those 505 cell cycle genes, I ranked them by feature importance to the label, and using from that set, using the top 3, 4, 5, all the way to top 50, which is shown here, and repeating this multiple times, conducting tenfold cross-validation, and computing the sum squared error of classification here. So you can see the error is high where it's only 3 genes, and it drops down when it's coming to like around here, 16, 17 or so. And the optimal number of genes that will be used, I'll show you later, that I'm using, as opposed to that 40 genes of the paper, is the minimum, finding the minimum of these measurements, error measurements. So here's so that in a way is a supervised method, right? So this linear SVM that I'm using to be able to do this is a supervised method. And you can see that in supervised methods is also biased by the normalization of quant norm, rank-based norm, or log math compared to the log scaled count, which is the first one. And you can see that, just look at holistically the change in the number of genes that could come in and how it's dramatically affects these changes. Because it's really difficult to be able to show that there are actually different genes found by each of these approach that I took, here's the Scialdone et al. 40 genes down here. And the yellow is showing the G2s, the pink is G1s, and the blue is the S's. And log scale counts, rank normalized, quant normalized, and log mav normalized, and for each one. And so you can see that, in fact, the gene sets that I found to be able to be best classify them with a linear classifier is different than the original 40 genes. The limitation here could be that they overfit to the training, which means that using these gene sets, we could overfit to the training set and not be able to generalize them to a training set, which is what I will show you that it's not the case. So, an overview of what the classification is happening. Like I said, we're classifying these cell cycle labels in these two data sets. First, we reduce them to the 405 cycle genes. As I said, normalization, if done, we can just do it at this stage. Then reduce it by either that 40 gene set or that top end gene set that I just described, depending on which one that we use. And so now we have these narrow gene expression matrices, which I can create training and validation sets from. And in a 5-fold cross-validation, where 4 folds is held and inference is done on this one, and it's rotated so that every fold is held once back, and training is done on first fold and then the other one. And for external validation, where we're testing the generalizability of the classifier, we're classifying on 4 folds from the one data set, and they're inferencing on the other one. So, to make the final call of the classifiers, we have to, because there's some stochasticity associated with the classification in that gradient descent algorithm that tries to find the local minima, and a few other features in the actual framework, running it one time will not produce a good result. We have to run it multiple times and get I think it's better to do it that way, and to get a probability associated with each class label, and then make the final call on that. And that's done in this sort of, like I said, the k-fold, where the validation is held, and then there's training is done on the rest, and that's rotated across. And this whole thing is repeated, in this case, 25 times. Now, to compare models, I'm going to quickly introduce the F1 measure, which is a harmonic mean between precision and recall. It's a good measure because in rare cellular subsets, we are worried about what measure to use. If, for example, accuracy would be bad, you can classify something as 99% accurate, but reality is you may not have done a good job if the rare cellular subset is like 1% itself. So, but F1 is actually a good measure that kind of balances, it's a combination measure, so it balances these two, and it's fairly well used in literature as well for these sort of purposes. So as I said, the first aim, part 2, was to be able to evaluate and benchmark the performance. So here is, because it was rank normalization, here is the RTL framework, the baseline version and its extended version compared to a similar linear SVM here. And this figure on the side here is what Scialdone et al. Is directly from their manuscript. And what I like to show is actually the more important the thing to look at here is the red lines, which is the macro average F1 scores across. And so in that regard, because in reality, you have to compare each cell cycle label to each cell cycle if we're making comparisons. But as a whole, if you're making an average call on which perform better, let's look at just the red lines. And we can see that on average, with the rank normalization, we didn't outperform Scialdone et al. But if we look at, did we outperform a linear basic SVM? Yes, we did, with both of them, but with the baseline a little better. Now, what about the other normalizations that I talked about? So compared to, to each other or to that linear SVM, we can see that the log math actually produced the highest of all of them. And even if you want to have a different kind of a normalization, the quant norm, it produces higher normalization than the rank norm with the RTL framework. So again, there is some effect from the normalization on the classification. Right, so the log map also outbeats any of them. But don't get too excited on that. There's a reason that that's actually not the right thing. But I just want to highlight that if this log map wasn't here, the next highest one, which is very equivalent to one of the higher points here, just maybe slightly off of this one, but their method, the actual method that they talk about is this pairs method. So these are not the rest is not their developed method. They developed this pairs method. But regardless, on the just log scale counts, without any normalization, we produced a pretty well classification F1 score, if that log map was in there, but log map beat them all. Now, with the top N genes, so because like I said, the 40 gene signature is good, but not optimized for a linear classifier, so these top N genes are, and we can see that throughout, they all produce higher and much higher than those scores. So again, like I said, the limitation is how well does this transfer, because this internal validation that we're doing is just on one data set being split apart. And one part is being held out for that testing and then rotated. So part 2 is about generalizability, where you're learning on one data set, you're inferring on the other one. And again, we start with the basic one, where we're looking at just rank normalization. We're doing a direct comparison. We don't beat their score. The highest one, in fact, they achieved was with the PCA method that beats their own method. But and there are reasons a little bit for that. But regardless, if you just compare it to a linear SVM, we outperform a linear SVM by doing a transfer learning as opposed to supervised method, which is the SVM linear method. Then compared to the other normalizations, now look at the log map. It didn't generalize. So that's why I said, hold your excitement there. But if you ignore this guy, you can see that the next highest scores are either on the log count or potentially the quant norm. But in general, this one produces, is one of the next highest one as well. So now the, compared, well, that's, I was just saying that here, that still PCA beats that. Oops. But the top N genes. Now, the generalization is not perfect, but you can see that the trend with the log map still remains, but you can see that some of these are producing higher classification, even generalized, onto this other new data set. So the top N did not overfit to training those signatures. So regardless of that, you can see that what's the highest achieved F1 score by these methods is on the log counts, followed by the quant norm with the extended version. So now to the actual point, which is the cellular subsets, to be able to evaluate those. Now, in all honesty, as I showed, that these cells were not rare by any definition. And in my manuscript, in my dissertation, I kind of am defining them less than 5%, but based on other work, obviously. But here, these are not that in that ratio. So how do we get that? And that I did in silico resampling. There's several different methods that we can do. The simplest one is sampling the negative class. Basically, you bring in all the positive class that you want. For example, in this case, the G2M cells, and you just randomly, without repeat, with repeat, you take from the negative population to increase the negative samples. So now the marginal frequency is rare. And again, with the same scenario where we're doing the n repeated k-folds. So in the context of rare, I'm showing only one at 5%, one at 1%. Less than 1%, it wasn't really good to go below that with this data set. So I've tested all the way down to 1, but I'm showing 5 and 1 here, and comparing baseline extended, and then with the parameter transfer I talked earlier, and without the parameter transfer. So first, without parameter transfer, we see that in general, the extended framework works better than the baseline, and they both perform better than just doing a basic linear SVM supervised. However, when the parameter transfer comes in, to make that inference on narrowing that space, we see that the baseline performed better as opposed to the extended. And there are a few reasons for that, mostly because potentially the extended version is over-smoothing, so it's changing the curve and so the area behind it and where the estimation line falls. That's where I've kind of found the root of it for now. But yeah, in general, so the idea would be if you're looking for rare cellular subsets, you can do with parameter transfer with the baseline version should get you the highest achieved potential F score. But again, it really depends on the data itself. I mean, this data had some noise and had some, as I showed earlier, there was some overlap in the labels. So, but it's a real case applied work, so we don't expect 100%. So as a summary, what I showed was that the RTL framework developed, which contains the baseline and extended versions, performs better or equal to the Scialdone version, Scialdone reported F1 scores. The extended version performed better than the baseline version relative to the F1 scores when you're going across data sets in generalizability. And in terms of rare cellular subsets down to 1%, the baseline with parameter transfer performed the best out of the other settings. That's and in terms of doing this with other types of data, in my dissertation, I have on a large data set, flow cytometry data set, shown that it works there as well as on obviously a single cell RNA-seq data. So in terms of potential future work, as I said, the binary classification, we can go do better, do multi-class cell population identification, potentially through networks or tree-based methods that this transfer learning can be applied to find actual and applied to use this to find causes of health and disease and find or prophylactic protection. Use this framework to be able to find kinetics of gene expression or temporality between phenotypes, pseudo-temporal ordering of the cells depending on, again, like cell cycle and putting them in different lineages, or finding these regulatory networks or these cellular moietisms that exist in us to be able to classify them as separately subpopulations of cells and be able to study them. And with that, I want to thank Mariana, who's sitting in the back over there, my partner who for all these years has been supported my emotional needs and being to, and the PhD work can be a roller coaster sometimes. And of course, with the help of Kenny, who's been mostly an emotional dog, support dog. And I also want to acknowledge my mentorship of Dr. McWinney and Dr. Lewinson and Dr. Gold, as well as my committee, which includes Dr. McWinney and Dr. Lewinson, as well as Dr. Mooney, Dr. Zhang, Dr. Lind, and the external reviewer who's not here, Dr. Ady. And also I'd like to thank Debbie Lewinson, Dr. Lewinson, the other one. That's how I define it. As well as Dr. Lederes. Especially I want to thank the DMI staff and other members in the DMI student cohort and all not just my cohort, but all of them throughout. It was really nice to know you guys, and I've really been influenced by all of you. And with that, I take questions. Thank you very much. Yes, Eric.
Hey, I just had a nice talk.
Thank you.
I had a quick question about the top N genes. So it looks like you were doing a lot of repeated cross-fold validations. So I'm wondering if you looked at whether or not the top N genes you identified had like a consensus where you identified the same genes over and over again, and if so, did they match with what you had in the 40 set?
Yeah, I think,
or was it kind of like a random grab bag and they never really overlapped?
The bag that I started with was the 405 cell cycle genes that they came with. But if I can go back to the little nice, at first this was not very liked very much, but I think it still makes sense. I changed a little bit, so hopefully it doesn't make the point that I was trying to make. So the Scialdone et al. Gene set is just these 40 genes. And the gene sets that I found, there is some overlap, like some of these genes, whatever they are, there is some overlap in some of them, right? But there are some unique gene sets that are, or genes in those sets that are not in the, or in here.
Okay.
How they relate to biology and stuff, that, like the biology of cell division, that's another, a little out of the scope, but that's another level to investigate this to further. So actually, do they make biological meaning, these sets? Yes, sir.
Between the three different categories of phases, it seems like the G2M phase and the G1 phases of cell subsets were always identified more easily through the algorithm with the higher F1 score. So is there a generalizable characteristic of a data set that will work better with your algorithm versus the S phase that was almost always the lowest?
Right. In Scialdone et al. That published, they actually say that specifically, that that was also the hardest one to classify. So that is a similar finding. I think that S phase in general is a complicated state that the cells are in, that there's a lot of transcription going on, and that makes it a little bit more difficult to narrow them. And there's a lot of overlap, like between the other cell cycle phases, but G1 phase and the G2 and M phase combined had, even in their work, showed the best signature in classifying. But maybe specifically designed for the S phase cells, but generally what this is used for in a pipeline of RNA-seq pipeline, for example, is to remove cell cycle as a confounding factor. So when you want to look at other biology, who cares about the cell cycle? But it's important in this case because I'm actually using it to classify them. Does that answer the question?
So I guess I would have hypothesized if there were more unique genes expressed during S phase that it might be more readily identified as a unique cell subset, but it seems like it might be the opposite.
Yeah, I would say more examples of S cells as well. Like just if this was a large data set, I might have been able to maybe say, oh, there's two classes of them maybe, and that. But with the number that is there, I can't do that. Shannon, yes.
So in the gene expression literature around classification of signatures, there was a pretty important paper that came out a couple of years ago that showed that random signatures would do just as well. And that's obviously a slightly different paradigm than what you're doing with the transfer learning, and I'm just curious your thoughts on what the right assessment might be. You know what I'm saying? Similarly, right, for transfer learning, are there certain checks that we should be doing, especially to be cautious about what is actual, because you know in your last slide you're giving all these different applications that it could be used for, and a lot of those are actually linked to the idea that the classification itself is meaningful.
Right.
And the paper that I'm referring to would actually argue that it's just the correlation structure. There's nothing specific in a lot of these signatures about the actual numbers of the signatures. It's really about the fact that they have a correlation structure that is important for the outcome or the phenotype of interest. So do you have any thoughts on that for kind of in a future direction, the type of computational things we might need to assess for transfer learning?
I think that other omics data have to be integrated. It's like partially thinking about genes that actually make meaning, like I said, biological meaning, but like integrated through other omics, like network from like either gene ontology or from like phenomics itself. That you can bring in some of those potential network-based omics that integrate into this to be able to classify or create these gene sets that are more biologically meaningful. Is that what you're trying to say? That just not just a correlation, but it's actually biology?
I'm just asking your opinion, I think. So you think that if you bring in other omics data types, that would help? Because I think that what I'm trying to get at is, given what you've learned, which is a lot about transfer learning, and especially some of the concerns around some of these different methods, in terms of the emphasis that people put on the actual members of the classifier, or the predictors. I guess I was just wondering if, so in your mind it would be the other data types that would actually help you?
I mean, I don't think, as far as I've tested, I don't find that transfer learning is unique in its way that we find gene sets for. in fact, the way I found them was through a supervised method, which then I'm using as a transfer. But yeah, I think that non-specific to transfer learning, but to improve the meaning of these gene sets, I think that other omics should come in. If that doesn't answer your question, or does that?
It's okay, we can take it off.
Okay, if any other questions in there? All right, with that, thank you very much, everyone, and thank you for being here. And we ask the public to leave me in the battle alone.